Genetic counselors. Genetic counselors can provide information to prospective parents concerned about a family history for a specific disease. Using family histories, genetic counselors help couples determine the odds that their children will have genetic disorders. For a growing number of diseases, tests are available that identify carriers and help define the odds more accurately. Fetal testing. There are several technologies available that can help provide insight into potential genetic defects. Parents can be tested to see if they are carriers of a certain allele. Amniocentesis. Amniocentesis is a procedure performed between weeks 14 and 20 of pregnancy in which a needle is inserted into the mother's uterus and some amniotic fluid is extracted. That's why it's called amniocentesis. Fetal cells found within this fluid can be tested for genetic disorders. So in the diagram we can see that a needle is inserted into the amniotic cavity, the fluid is extracted, and then we centrifuge the fluid to separate out the cells, which are here, and then the cells are grown on a cell culture, and then we can later look at the chromosomes in a karyotype. Click here to see a video about amniocentesis. Chorionic villus sampling. In chorionic villus sampling, a sample of the placenta is removed and tested. This is as opposed to the amniotic fluid in amniocentesis. Results can be obtained within 24 hours, therefore much quicker than amniocentesis. Another advantage is that it can be performed as early as week 8 of pregnancy. Both amniocentesis and CVS pose some risk of serious complications. Therefore, they are generally only done in situations where the risk of a genetic disorder is relatively high. Karyotype. The samples obtained via amniocentesis and chorionic villus sampling are used to produce a fetal karyotype. Remember from our previous unit that a karyotype is a photographic inventory of chromosomes. In order to get this picture, the chromosomes are isolated, stained, and examined under a microscope. The pictures of the chromosomes are taken, and then the pictures themselves are arranged in order of decreasing size. So chromosome 1 is the largest, and chromosome 22 is the smallest. Fetal karyotypes are used to diagnose chromosomal abnormalities before birth. In this karyotype, it's a human female showing 23 sets of homologous chromosomes. We know it's a female because the karyotype shows 2 X chromosomes. Ultrasound and fetoscopy. Other techniques, such as ultrasound and fetoscopy, allow a doctor to examine a fetus directly for anatomical deformities in utero. Ultrasound imaging uses sound waves to produce a picture of the fetus. The procedure is non-invasive and has no known risks. Fetoscopy involves the insertion of a long, thin tube containing a scope and fiber optics into the uterus. It can provide highly detailed images of the fetus, but does have some risks involved. Fetoscopy can also be used to do fetal surgery in utero. Click here to see a video on fetoscopy. Common genetic tests. Some genetic disorders can be detected at birth by simple tests that are now routinely performed in most hospitals in the United States. One of the most common tests is PKU screening. Phenylketonuria is a recessively inherited disorder that occurs in about 1 out of every 10,000 births in the United States. Children with this disorder cannot properly break down the amino acid phenylalanine, which may lead to developmental and/or intellectual disabilities. If this is detected in a newborn, the baby is placed on a special diet low in phenylalanine, and this will usually prevent developmental disabilities, because it's during those first few years that the brain really develops.